Hey y'all, Dr. Avinash here, your host, your mentor. Today, a chapter called as the Respiration in Plants. Super important chapter. Grab your textbooks, your highlighter, markers, post-it notes, and let's make this chapter super duper easy, super duper simple. Here we go. As usual, we are not going to start from the beginning of the chapter. The content given at the start might put you off. Your brain might go into a brain fog. So where do we start with? We start here, glycolysis. This chapter is about how plants break sugars and generate energy out of it, energy in the form of ATPs, right? So this chapter will have the following things: glycolysis, which of course is breakdown of glucose. Then we have a thing called a Krebs cycle, and then electron transport system. These three are going to be the heavyweights of this chapter. You must know these three concepts, okay? The rest of the things will follow. So let's start from the beginning. What exactly is glycolysis? Well, the word comes from a Greek word for glycose, sugar, and lysis for splitting. So literally glycolysis is sugar splitting. Who discovered this pathway? The scheme of glycolysis was given by three scientists, Gustav Embden, Otto Meyerhof, and J. Parnas. And that's why this pathway is rightly called the EMP pathway, the Embden-Meyerhof-Parnas pathway. Where does this pathway actually happen? This pathway occurs in the cytoplasm of the cell. And who has cytoplasm? Well, all living cells have cytoplasm. So it's literally present in all living organisms. And that's why glycolysis is common, right from bacteria to the biggest trees and the biggest animals. Everybody has glycolysis, including the anaerobic organism. As a matter of fact, in anaerobic organisms now, only glycolysis is present. It is the only process of respiration in anaerobic plants. Simple. What exactly happens here? Array, the glucose now undergoes partial oxidation, not complete oxidation, partial oxidation to form what? Two molecules of pyruvic acid. Okay. Now, question. Where do these plants get glucose from? Array, glucose, it happened in photosynthesis. Now, it was stored in the form of sucrose. So in plants, glucose is derived from sucrose. Okay, and how did we get sucrose? Well, we made it at the end of photosynthesis. At the end of photosynthesis, we taught you glucose comes, but the plant store glucose in the form of sucrose initially. Or if it is a long-term storage, they can even form something called a starch. So that is what is written here as from storage carbohydrates, so starch basically. Okay, now important for us to remember the starch or sucrose, because our pathway actually starting at sucrose, will start with sucrose. So if you take a molecule of sucrose and you add an enzyme on it, this enzyme is called as invertase. Okay, that enzyme converts sucrose into one molecule of glucose plus one molecule of fructose. So basically sucrose now is a 12 carbon molecule. It breaks down into 6 carbon and 6 carbon glucose and fructose each. Easy. So sucrose is converted into glucose and fructose by the enzyme invertase. And these glucose and fructose, they enter the glycolytic pathway. We will now first have a look at glucose. Understand glucose, then we'll talk about fructose. So before we read this paragraph, I want you to go through this particular flow chart with me. Glucose is converted into glucose 6-phosphate by a special enzyme. This enzyme is given here. It's called hexokinase. Okay, so the glucose is converted to glucose 6-phosphate by an enzyme called as hexokinase. This process requires a molecule of ATP. So you spend a molecule of ATP before you get ATPs out. It's like business. You invest first, then you get it back. So you invested a molecule of ATP, it became ADP, and that phosphate now, ATP adenosine triphosphate, it became adenosine diphosphate. So where did that phosphate go? Here. It went on the 6th carbon. See, glucose 6-phosphate. Then a simple isomerization takes place. and it forms fructose 6-phosphate, 6 carbon compounds. Very simple. Then another kinase works to convert fructose 6-phosphate into fructose 1,6 bisphosphate. Now, why bisphosphate? Why not diphosphate? Array, see, look, if there is one carbon, and on that carbon you attach two phosphates like this, you call it a diphosphate. Getting it? But here, if you notice, there are 6 carbons of fructose. The phosphate is attached to the first and to the sixth. Now, because the phosphate is attached to two different carbons, it's appropriate to call it bis. Getting it? So, di and bis has its significance. Okay, so fructose 1,6 bisphosphate. By this time, you have invested 1 plus 1, 2 ATP. You're already in the negatives. But now, the real fun part starts. The fructose 1,6 bisphosphate immediately breaks into 3 carbon molecules. So 6 carbons are divided into 3 carbon and 3 carbon. The two products are glyceraldehyde 3-phosphate, also called as PGAL, and dihydroxyacetone phosphate, also called as DHAP. Simple, simple. So literally speaking, you get a split product. 3 carbon, 3 carbon, 3 carbon glyceraldehyde 3-phosphate or phosphoglyceraldehyde PGAL and dihydroxyacetone phosphate or DHAP. Simple, simple. Now, let us keep DHAP on hold. Okay, so what we are going to do right now is this molecule here we are not going to use. Okay, let this be at hold, and I'm going to use only these 3 carbons for the time being. For the time being, this glyceraldehyde 3-phosphate is converted into 1,3 bisphosphoglyceric acid or BPGA. Bisphosphoglyceric acid. Now, what they have not shown you here is where is this carbon coming from the phosphate coming from at carbon number 1. Well, it's basically an inorganic phosphate. You're not wasting any more ATPs now. No more ATPs wasted. 2 invested, more than sufficient. Now is the time to get ATPs. So glucose became glucose 6-phosphate, became fructose 6-phosphate, became fructose 1,6 bisphosphate. This 6 carbon fructose 1,6 bisphosphate splits into 2 3 carbon compounds, PGAL and DHAP. Let us keep DHAP on hold. Let's start with PGAL. The 3 carbon PGAL become 1,3 bis PGA. Bis PGA. During this time, oxidation takes place. Oxidation. See, when you are doing oxidation, you remove hydrogen, right? Right. So if you're doing oxidation, somebody else has to get reduced. Barabar, na? Who is that somebody? It's an energy molecule called as NAD. Not energy molecule. Let's not call it an energy molecule. Let us call it, what can I say? It's kind of a check. If ATP is the cash currency of your cell, NAD is the check of your cell. Getting it? So we are going to create a reservoir of energy. We can't use it directly, but we have enough ATPs inside, hidden inside. So literally speaking, 3 ATPs are hidden here. So you take an NAD plus actually, and take that hydrogen from PGAL and convert NAD into NADH plus H plus. Getting this idea? You oxidized glyceraldehyde 3-phosphate to 1,3 bisphosphoglyceric acid and at the same time reduced NAD to NADH plus H plus. Getting it? How many NAD did you get? 1. Getting it? As of now, we are ignoring this 2. We get 1 3 carbon PGAL converted into 3 carbon 1,3 bis PGA. Then you will convert this into phosphoglyceric acid, 3 phosphoglyceric acid or 3 PGA. Listen, important. 1,3 bis PGA became 3 PGA. Where did that phosphate on carbon number 1 go? It went to ADP. See. It went to ADP and now you got an ATP. This is the first earning you have directly. When you get direct ATP in any reaction, you call that as substrate level phosphorylation. Simple, simple. So this was our first substrate level phosphorylation. You got one ATP here. So till now you have one check called as NADH plus H plus and one cash molecule of ATP. The reaction will be called as substrate level phosphorylation. Getting it? Now the 3-PGA became 2-PGA. Right now we are just ignoring these numbers. We got 2-PGA, 2-phosphoglycerate, 2-PGA. Nothing has happened. Phosphate has gone from carbon number 3 to carbon number 2. Okay, so there is no ATP ka jhanjat here. Now, remove a water molecule and you get PEP, 2-PEP, phosphoenol pyruvate. Ignore this. Phosphoenol pyruvate, and phosphoenol pyruvate will eventually form, you know what? Pyruvic acid, a 3-carbon pyruvic acid. Let me write this as PA, pyruvic acid. Getting it? Simple, simple, easy. Glucose to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6 bisphosphate to DHAP and PGAL. We kept the DHAP on hold. We are not talking about this as of now. The PGL became 1,3 bis PGA, 3 PGA, 2 PGA, 2 PEP and pyruvic acid. Important MCQ points. This is another substrate level phosphorylation. You got a +1 here. Simple, simple. Also, the points for the champions, the future doctors. Look at this. In this step, what you essentially did was dehydration. You removed a water molecule to convert 2-PGA into 2-PEP. I hope things are clear. And if they're not clear, you know what you will do? You will comment in the section below so that I can solve your doubts. I'm here to help you. Okay? Now, once this is done, remember, we have kept a molecule of DHAP on hold here. It's still there, right? So let us convert this DHAP, dihydroxyacetone phosphate, into PGAL. Getting it? See, you can convert that. And the remaining path, now you will do it one more time. So if you do it one more time, you know what happens? You get one more NADH plus H plus. So now we have total of 2. And then you get 2 ATP in the first substrate level phosphorylation. And then you have 2 ATP in the second substrate level phosphorylation. Getting it? At the end of glycolysis, now let us zoom out and check what we have done. We have invested 2 ATPs. So you are minus 2 ATP at the start. And then at the end, you got 2 plus 2. That's 4 ATP directly. And you also got 2 molecules of NADH plus H plus. And as I said, this is equal to 3 ATPs. So if this is really, really true, how many ATPs do you have from NADH plus H plus? 3 2s are 6 ATPs, right? So how many ATPs did you earn out of this entire process? 6 plus 4. 4 from substrate level phosphorylation here, and 6 indirectly from NADH plus H plus. So you got a gross of 10 ATPs. Getting it? Array, but this was business now. You invested 2. So don't you have to minus that 2? Yes. So when somebody says, what is the net gain? The net gain would be 10 minus 2, that is 8 ATP. Simple. So if you do aerobic respiration, okay, now words are important. If you do aerobic respiration, only then the gross gain of ATP is 10 and the net gain of ATP is 8. Why did we say aerobic only? Later we will check out. Okay, but as of now, are we clear with this particular pathway? If yes, give me a thumbs up and write in the comment section below, yes, I am clear with glycolysis. So I know that I have been of some help to you. Now that you are done with this understanding, let us go through the entire pathway. Both glucose and fructose can enter this pathway. They have written, Array, but they have only given glucose. Where is the fructose? If they have not given, it is our duty to write it. So I'm going to clear this off so that we can add something here. Let's start writing the additional things. If you start with the molecule of fructose, which is a 6-carbon compound, you add a molecule of ATP to it, just like the this one here. You convert that ATP into ADP and form, guess what? Fructose 6-phosphate. The enzyme is the same. The only difference is to reach this step here, glucose did it in 2 steps, glucose to glucose 6-phosphate to fructose 6-phosphate using 1 ATP, correct? And fructose did it in 1 step. Fructose became fructose to fructose 6-phosphate using 1 ATP. So there is no change in the number of ATPs or NADH plus H plus. Yeah. The only thing that changes is steps. Glucose to glucose 6-phosphate to fructose 6-phosphate. And if you start from fructose becomes fructose 6-phosphate. The end. Enzyme is also same. You don't have to remember anything extra. So glucose and fructose are phosphorylated, ATP added to give rise to glucose 6-phosphate by the activity of enzyme hexokinase. And then the glucose one will get isomerized to form fructose 6-phosphate, getting it. The remaining steps are same. Simple. So the total number of steps were 10. A, MCQ. Glycolysis has how many reaction? 10 reaction, chain of 10 reaction. Each is under a different enzyme. Correct? And where does it take place? In the pyruvate. And what does it produce? It produces pyruvate from, where does it take place? In the cytoplasm. And what does it produce? Pyruvate from glucose. Simple. We have consciously noted down that 1 ATP is spent from glucose to glucose 6-phosphate or fructose to fructose 6-phosphate, whatever. And 1 between fructose 6-phosphate to fructose 1,6 bisphosphates. You are down by 2 ATP. But then you have 2 substrate level phosphorylation steps through which you will get how many ATPs? Total 4, correct? And then you have 1 NADH plus H plus last step, 2 NADH plus H plus are generated, in which you will get 6 ATP. Clear? So now that we are clear with this, let's read and finish it. ATP is utilized at 2 steps. First, in conversion of glucose to glucose 6-phosphate. You know this. Second, in the conversion of fructose 6-phosphate to fructose 1,6 biphosphate. Clear? Awesome, simple, amazing. You guys rock. Fructose 1,6 biphosphate is split into 2. DHAP, that's dihydroxyacetone phosphate, and 3-phosphoglyceraldehyde or PGAL. Hey, how awesome these people are. Here they write 3-phosphoglyceraldehyde. But here, you know what they wrote? Glyceraldehyde 3-phosphate. But you people are smart. You are not going to make that mistake. Glyceraldehyde 3-phosphate is the same as phosphoglyceraldehyde. It's the same thing. Which carbon is phosphate attached to? Third carbon. It's written here, na? Now, in this step, what was the speciality you remember? NADH plus H plus is formed from NAD plus. And PGAL is converted into bis PGA 1,3 bis PGA. Now, during this process, MCQ, 2 redox equivalents are removed in the form of 2 hydrogen atoms. It is removed from whom? From PGAL. And given to whom? It is transferred to a molecule of NAD+. So what exactly happened now is PGL got oxidized. Barabar. And if you remember, we also added an inorganic phosphate. So this is what they're mentioning here. See, and with inorganic phosphate, it gets converted into 1,3 bis PGA. Simple. What was the next step? Remember? 1,3 bis PGA becomes 3 PGA. It's an energy yielding process. And where do you trap the energy? ATP. And one more ATP is made. Conversion of PEP to pyruvic acid. And now you know the calculations. Easy. So at the end of this entire process of glycolysis, what did you achieve? Pyruvic acid. What happens to pyruvic acid? It depends on what the cell needs. Easy. There are three options to pyruvic acid. And we are not discussing it right now because it will distract us from our focus. So, part number 1 of this chapter is glycolysis. Start here, 12.2. This is given on page number 155. Finish page 156. Done. Now what to do? Now you flip a page ahead to page number 158 and come to a concept called as aerobic respiration. Where does aerobic respiration take place? In eukaryotes, aerobic respiration takes place in the mitochondria. It requires oxygen. Because it requires oxygen, I am going to call it aerobic. Aerobic respiration is the process that leads to a complete oxidation. Glycolysis. Incomplete oxidation. Aerobic respiration. Complete oxidation of organic substance like glucose, pyruvic acid in the presence of oxygen, and releases carbon dioxide, water, and large amount of ATP. Very common in higher animals and plants. Actually, higher organisms. That's why they have written higher organisms, not in plants, higher organism. Even you and I are doing Krebs cycle. See how awesome it is. So, for aerobic respiration to take place, you know what is supposed to happen? At the end of glycolysis, what is formed? Pyruvic acid. Where is the pyruvic acid formed? Glycolysis happens in the cytoplasm. So pyruvic acid will also be formed in the cytoplasm. Na? Correct. But Krebs cycle or the aerobic respiration happens inside the mitochondria. So the number one thing that should happen is the final product of glycolysis, the pyruvate, has to be transported from the cytoplasm into the mitochondria. Simple. Once inside, pyruvate, the 3 carbon, will be completely broken down. All 3 carbons will be removed in the form of carbon dioxide in a step-by-step manner. Stepwise removal of all hydrogen atoms and carbon dioxide is out. We want the hydrogen. We love the hydrogen. Hydrogen is where the energy is. So you remove the hydrogen and you throw away carbon dioxide. And in that process, you pass on the electrons that we got from the hydrogen atoms to oxygen. Okay. So, हिंदी में, just to make things simpler, करना क्या है? कार्बोहाइड्रेट से हाइड्रोजन निकालना है, तो कार्बन डाइऑक्साइड भाग जाएगा, एंड हाइड्रोजन से इलेक्ट्रॉन निकाल के किसको देना है? ऑक्सीजन. ठीक है. इस पूरे प्रोसेस के वजह से क्या होगा? एटीपी बनेगा. नाउ इन इंग्लिश, व्हाट्स द पर्पस? Take a molecule of carbohydrate like glucose, take all the hydrogens. So what is left behind? Carbon dioxide. Throw it away. That hydrogen has to be split into H plus and E minus. That E minus is the key to making ATP. And who will take that electron? Eventually, the electrons will be passed to molecular oxygen. Molecular oxygen. Easy? Cool. Now, the electron transport happens in the mitochondrial membrane, inner mitochondrial membrane. The second process that we discussed happens on the inner mitochondrial membrane. Whereas the first process we discussed, which means the complete breakdown, it happens in the matrix of the mitochondria. So for people who are still a little bit confused, if this is my mitochondria, and you remember the mitochondria has these crystal-like structures from the chapter Cell, the unit of life, Krebs cycle happens here in the mitochondrial matrix, whereas the electron transport chain happens here in the inner mitochondrial membrane. Krebs cycle happens here. Oops, sorry. The Krebs cycle happens here in the matrix, and the electron transport chain happens here on the inner mitochondrial membrane. Cool. Now, think of it this way. Pyruvate was formed in the cytoplasm, right? That pyruvate wants to go inside the mitochondria. Ticket lena padega or fees bharna padega. The pyruvate has to take a ticket, coenzyme A, and it has to pay the price. The price is hydrogen. And if you remove hydrogen, you know what comes out? You know you are smart. If you're removing hydrogen, what comes out? Carbon dioxide. So if you give two hydrogen, you have to give away a carbon dioxide. So as you can see in this yellow box of us, pyruvic acid gets a ticket called as coenzyme A. This is its ticket to go inside the mitochondria. But in that process, the enzyme pyruvate dehydrogenase removes the carbon dioxide. A, decarboxylation. कितना मस्त नाम है ना? Decarboxylation. Removes the carbon dioxide. Makes a molecule of NADH plus H plus C. H plus K. From where? From pyruvic acid. And the coenzyme A got attached to the remaining two carbons. The remaining two carbons form the acetyl-CoA. How beautiful is that? The 3-carbon wala pyruvic acid gave away 1 carbon dioxide to buy the ticket called as coenzyme A. It also paid in the form of H2. So it lost the carbon dioxide. So only 2 carbons are now remaining with acetyl-CoA. This carbon dioxide will go away out into the atmosphere, and you got one more check. Hey, how can one more check come? When you did glycolysis, at the end you had 2 pyruvic acid, na? So in reality, for every molecule of pyruvate, you get 1 NADH plus H plus. But for every molecule of glucose, you will get this twice. Hey, getting it? Simple. Like, I hope things are clear. So pyruvate, which is formed from glycolytic catabolism. Glycolysis का breakdown of carbohydrate in the cytoplasm, it enters mitochondrial matrix and undergoes oxidative decarboxylation. Oxidative decarboxylation. ऐसा मतलब क्या? अरे NADH बना ना, so you gave away hydrogen. If you give away hydrogen, what has happened to pyruvate? It has undergone oxidation, and it also gave away carbon dioxide. So it has undergone decarboxylation. Beautiful word, super important. Oxidative decarboxylation. Remember this for your MCQs. So before it even enters into Krebs cycle, it has undergone oxidative decarboxylation by a complex set of reactions. And this complex set of reaction is catalyzed by our hero, the pyruvate dehydrogenase. Now, this is for the future doctors, the toppers of the NEET exam. Pyruvate dehydrogenase is requiring a cofactor. You know what is that cofactor? Mg. Look, here it is, given right here in front of our eyes. Remember it. By heart it. So, pyruvate dehydrogenase requires too many coenzymes. Too many coenzymes are required and a metal. But the coenzymes that are important for our syllabus are NAD plus and coenzyme A. So in this entire process, if you start from one molecule of glucose, you got 2 pyruvic acid, correct? So this is 2 pyruvic acid. You require 2 coenzyme A, you require 2 NAD plus, and you will get 2 molecules of acetyl-CoA, 2 molecules of carbon dioxide, and 2 NADH plus H plus. Exactly what we were discussing. See how beautiful it is. During this process, 2 molecules of NADH are produced from the metabolism of 2 molecules of pyruvic acid produced from 1 molecule of glucose. How simple is this chapter? Once you understand the basic concept, you can't go wrong, bachalo. Easy it is. Then the acetyl-CoA enters a cyclical pathway called as the tricarboxylic acid cycle. Acetyl-CoA enters a cyclical pathway called as the TCA cycle, the tricarboxylic acid cycle, more commonly called as the Kreb cycle, because who discovered it? Sir Hans Kreb, who first described it. Now, before I start with Krebs cycle, understand this reaction is also known as the link reaction. Okay? Link reaction is not a part of Krebs cycle. Krebs cycle starts later. Look They have mentioned it specifically. The TCA cycle starts with the condensation joining of acetyl group with oxaloacetic acid and water to yield citric acid. The first stable compound of TCA cycle is citric acid. Somebody synthesized citric acid, so the enzyme must be called citrate synthase. Easy. But it's not citrate-CoA, na? You started with acetyl-CoA plus oxaloacetic acid and water. But you got only citrate, which means a molecule of CoA is released. You know what is the other name for tricarboxylic acid cycle? It is also called a citric acid cycle. It's called as the citric acid cycle. You know why? Because the first product is citrate. Simple. Now, the remaining cycle we have to do from here. Oxaloacetic acid combines with water, which they have missed out here, and acetyl-CoA to form citric acid. Simple maths. Observe. Oxaloacetic acid, 4 carbon. Acetyl-CoA, 2 carbon. Citric acid, 6 carbon. Now, this citric acid, steps they have skipped. Citric acid is first converted into cis aconitate, which you may or may not remember, to isocitrate. It forms oxalosuccinate. And oxalosuccinate then forms alpha ketoglutarate. I hope I am adding to your knowledge and not creating confusion. But this is important. The citrate is converted or isomerized to isocitrate. Then it is converted into isocitrate to oxalosuccinate and oxalosuccinate to alpha ketoglutarate. Important for you. Here, a molecule of CO2 is released between isocitrate and oxalosuccinate. Okay? Between oxalosuccinate to alpha ketoglutarate, you get a molecule of NADH plus H plus. The alpha ketoglutarate is then converted into succinyl-CoA. Succinyl-CoA is converted to succinate. Succinate is converted to fumarate. Fumarate is converted to malate and malate is converted into oxaloacetic acid. Now, I want you to remember the following things extra, super duper important. Between succinyl-CoA, between alpha ketoglutarate and succinyl-CoA, carbon dioxide is released, NADH plus H plus is formed. Between succinyl-CoA to succinate, it is that GTP formation. This step is happening here. FADH2 is formed between succinate and fumarate. And NADH is formed between malate to oxaloacetic acid. I will be putting up on the screen the complete Kreb cycle pathway now. It is for you people to pause the video and copy this down for your information. Super important. Please copy this one. Okay. Coming back to our chapter. So this is how we said the reaction is catalyzed by enzyme citrate synthase. A molecule of coenzyme A is released. Citrate is then isomerized to isocitrate. Then remember, 2 carbon dioxide go away. Look, here and here 2 carbon dioxide went away. Na? So we call it decarboxylation. So this is followed by 2 successive steps of decarboxylation. And what did we get? Alpha ketoglutarate, and then we got, alpha ketoglutarate gave us succinyl-CoA. Barabar na? Isocitrate became oxalosuccinate to alpha ketoglutarate to succinyl-CoA. During this entire process, 2 carbon dioxides were released. Succinyl-CoA eventually will become oxaloacetic acid. Now, steps that they might have missed, but now you know because you have paused the video and copied it. Conversion of succinyl-CoA to succinic acid, GTP is made. A substrate level phosphorylation, super important. GTP is ATP, same. When you form GTP, you get ATP. GTP will be converted to GDP, and in return, ATP will be formed. I'll write it for you so it becomes easier for you. The GTP molecule becomes GDP, the diphosphate, and in return, the ADP molecule becomes ATP. The phosphate from here is added here. Getting it? Triphosphate became di, and di became tri. So coupled. They are coupled together. In a coupled reaction, GTP is converted to GDP with simultaneous synthesis of ATP from ADP. Clear? Are we clear with this line from the explanation given above? If yes, write down in the comment section, I understood the coupling of ATP and GTP. Okay, let's proceed. Also, there are 3 points in the cycle where NAD plus is reduced to NADH plus H plus, and 1 point where FAD is reduced to FADH2. Hey, what is FAD? We understood that NADH plus H plus is like a check. It can give you 3 ATP. FADH2 is also a check, but of a lesser value. It gives you 2 ATPs. Okay, so NADH plus H plus 3 ATPs and FADH2 2 ATPs. Now, so you started with acetyl-CoA, combined it with water and oxaloacetic acid, started citric acid, isomerized it to isocitrate, 2 steps of decarboxylation to form alpha-ketoglutarate and succinyl-CoA, then succinate to fumarate, malate, oxaloacetic acid. You made substrate level phosphorylation, GTP. You made 3 NADH plus H plus. See, 1, I'll just mark it again for you. 1, 2, 3 NADH plus H plus. You made 8 FADH2 in this entire process. But if you want this cycle to continue, the oxaloacetic acid has to be formed. Simultaneously, you also require NAD. See, without NAD, how will you get NADH plus H plus? Without FAD, how will you get FADH2? So, oxaloacetic acid jitna important hai na, the amount, to the level to which OAA is important, the same goes for FAD and NAD. Getting it? Both are equally important. Otherwise, you will not get NADH plus H plus and FADH2. So this reaction here sums up everything. But remember, this is for one pyruvic acid, not for a molecule of glucose. So if you're thinking about molecule of glucose, everything is into 2. And this is link plus Kreb. Okay? This is a combination of both. So at the end of link plus Kreb cycle, what is our let's say, total collection of ATPs? From glycolysis, what did you get? You got 2 substrate level phosphorylations, you got 4 ATP, correct? And how many NADH did we get? 2 NADH from glycolysis. From link and Kreb, for every pyruvate, you got 4 NADH plus H plus. So everything is into 2, na? So from here, you should get 8 NADH plus H plus, correct? And how many FADH2s will you get? 2 FADH2. And how many ATP should you get? 2 ATP. So if you total up everything, you know how much is the total, the net gain of NADH? NADH plus H plus will be 2 from glycolysis and 8 from here. That's 10. And if you remember, each NADH is 3 ATP. That would mean 30 ATPs are formed here. How many FADH2s? Total is 2. And you know that every FADH2 is 2 ATPs. So 2 into 2 is 4 ATPs. And how many ATPs did we get in total? Here there are 4, right? And here there are 2. So 6 ATP we got, right? 6. So this became a 40. Array, but did we not spend 2 ATPs? So what will be the net gain then? You lost 2 ATPs, you gained 40 in total. So 38 ATP is the best answer. Now, I know some of your professors have probably taught you about something called as a shuttle pathway that is used to transport pyruvic acid from the cytoplasm to the mitochondrial matrix before the link reaction. And sometimes that shuttle NAD is formed and sometimes FAD. Is it a part of your syllabus? No. If NTA asks you, will they ask you about the shuttle pathway? Never. If it is not in the textbook, the NTA will not ask it. So check out this 36 and 38 ATPs. Check out all this confusion about the shuttle pathway. Only focus on what is the information in the textbook. The textbook says 1 NADH2 H plus plus NADH plus H plus in the cytoplasm goes into the electron transport chain and gives us 3 ATP, so it must be giving us 3 ATP. We are not arguing about that. Okay? Not at all. It has to be 38. Okay? So don't get confused. Keep that clarity in mind. Hey, this is so easy, man. Glycolysis is easy. Kreb cycle is easy. Link reaction easy. Even the balance sheets are easy, right? Like now that you know things, they're simple. Yeah, they are simple. But don't you think we are missing out something very important? How did this NADH plus H plus or how did this FADH2 give me a molecule of ATP? How? Well, for that, you have to deposit that check in a special bank called as the ETS. The ETS is a bank that can cash your check. They take the NADH plus H plus, they take the FADH2 and convert it into ATPs that you can spend for your metabolic requirements. Getting it? So what exactly is ETS? I don't know why this seems to be difficult for so many students. Let me simplify it to a level where even a 10th standard kid will crack the NEET exam. Okay, let us break ETS down to a very, very simple level. I don't want any gassiness here. Just do me a small favor and observe what is a fact. In your textbook, what is given? Just observe that and you will understand ETS. Okay, let me make it even more simpler for you. This is a mitochondria. Oh, this became a circle. This is a mitochondria. Okay. And this mitochondria, of course, has crystal like this, right? And we said the electron transport chain is where? It is on the membrane, the inner membrane, right? Now, what you have to understand is, where is this membrane taken from? Imagine this membrane is here. This membrane, the blue colored structure is the inner membrane, has been taken from this part, this specific part here. What is outside the membrane? Can you tell me? Can you tell me by looking here? What is this space here? This is called as the intermembranous space. Correct, correct. This chamber here is the intermembranous space, right? This green thing that I'm highlighting. This is that green thing. Are we getting it? Getting it? Getting it? Clear, clear. Zoom in again. What is this part here? The yellow thing. It's the matrix, isn't it? It's the space inside the crystal. It's the matrix. It's the matrix. See, this part is the matrix. So this is inside the mitochondria, like inside the mitochondria, and this is the intermembrane space that we already know about. Are we getting it? Are we clear with the location of this entire process? Perfect, cool. Now, look at the diagram. I don't want to make you more confused. Look at the diagram and tell me how many complexes can you see? Well, I can see 5 of them. 1, 2, 3, 4, 5. I can see 5 complexes, but imagine somebody who can't really count properly has given them the numbers. So the complexes are 5, but the numbering is something like this. This is complex 1, this is complex 3, this is complex 2, this is complex 4, and this is complex 5. It's 1, 3, 2, 4, 5. Are we clear? Clear. I'm going to teach you the facts. 1, 3, 2, 4, 5. Acha, another thing I want you to very clearly observe, very, very nicely, it has been depicted, not at all difficult. Look at complex 1. Can you see it is protruding on this side of the membrane and it is also protruding on this side of the membrane. It is through and through. Look at this. It's protruding on this side of the membrane. It is protruding on this side of the membrane. Complex 3 is through and through. Getting it? Look at complex 4. This side of the membrane, this side of the membrane, through and through. Look at complex 5. This side of the membrane, this side of the membrane, through and through. Look at complex number 2. It is protruding on the inside, but it is not really coming outside. Look here. This fellow is kind of stunted. This fellow is stunted. It is not really a fully transmembrane complex. I hope you got it. We are just looking at the facts, Huh? How many complexes are there? Well, there are 5 complexes. 1, 3, 2, 4, 5. Simple. 1, 3, 2, 4, 5. Complex 1, complex 2, complex 1, complex 3, complex 4. Complex 1, complex 3, complex 2, complex 4, and complex 5. 5 complexes for you guys. 1, 3, 2, 4, 5. Out of which complex 1, complex 1, complex 3, complex 4, and complex 5 are transmembrane. Complex 2 is not transmembrane. Let us get the facts correct. The concept will take care of itself. Easy? Perfect. I want you to now focus on one very beautiful molecule which is found in all the organisms. All of them. It's as if this molecule is everywhere. It is ubiquitous. And because it's ubiquitous, everywhere present, let us call it ubiquinone. Where is this molecule? It is in the membrane. It is in the membrane. In the membrane. Here he is. Here he is. Are we clear? So membrane has 5 complexes. 1, 3, 2, 4, 5. 1, 3, 5, 4 are transmembrane, 2 is not. Within the membrane is a special electron carrier called as ubiquinone. Within the membrane, getting it? In the membrane. Okay? And look at someone special here. The cytochrome C. Where is the cytochrome C? It is on the outer side of the membrane. Getting it? It is on the outer side of the membrane, right? It is on the membrane. Ubiquinone was in the membrane. This was in. This is on. Now that you know how things are, let us understand the electron transport system. This is going to be beautiful, beautifully easy answer for us. Okay, really, really easy answer. Let's begin. The first check came into your account. NADH plus H plus. It doesn't matter from the cytoplasm or from the mitochondria. NADH came. NADH with its beautiful hydrogens, the power, the hydrogen. It came to complex number 1. Here, complex 1 took its hydrogen. Complex 1 took its hydrogen. Broke that hydrogen. So H2 became 2H plus 2E minus. Simple, easy, perfect. And now what this complex did was it started throwing some protons from this hydrogen itself, some from somewhere else. We are not interested. We are not going into unnecessary details to confuse ourselves. 4H plus were pumped out. How many H plus were pumped out? 4H plus were pumped out. What happened to the electrons then? The electrons were passed on through FMN to iron sulfur proteins, and the iron sulfur protein then gave it to ubiquinone. But you know now what happens when somebody gets electron. When somebody gets electron, they became reduced. So electron received to ubiquinone. Ubiquinone then gave that H and electrons to whom? complex 3. The complex 3 is basically something called a cytochrome. Cytochrome B, cytochrome C1, cytochrome B and cytochrome C1. That's why it's called a cytochrome B C1 with iron sulfur in it, of course. Iron sulfur in it. Complex 1 has iron sulfur, complex 3 has iron sulfur. Huh? Even this complex pushed out 4 hydrogen. You know why they can push out 4 hydrogen? Because they're freaking transmembrane. They can. They extend from here to here. They can pick up H plus from here and throw them out. They can literally take hydrogen from the matrix and put them out in the intermembrane space. Getting it? One more time. NADH came. NADH came. NADH plus H plus gave its hydrogen to complex number 1, which immediately splits it into electrons and H plus. The H plus is thrown out. The electrons are given to ubiquinone. The ubiquinone then passes it on to complex 3. Even as the electron passed through complex 3 cytochrome B, iron sulfur complex C1, cytochrome C1, even they push out H plus. The electrons are then given to cytochrome C. Remember, cytochrome C is where? It is on the outer side of the inner membrane. It is on the outer side of the inner mitochondrial membrane. This cytochrome C is an electron carrier. It carries electrons from complex 3 to complex 4. The complex 4 is unique. It doesn't have iron. It has 2 coppers. See, copper A and copper B. It has 2 coppers. And cytochrome A and A3. Now, because these two are taking electrons from cytochrome C, we will call them the cytochrome oxidase complex, cytochrome C oxidase. Getting the idea why they are called a cytochrome oxidase? Because they snatch the electrons from cytochrome C. And even they end up throwing hydrogen outside. Important for you to remember, complex 1 threw out 4H plus, complex 3 4H plus, complex 2 could only pump out 2H plus. Hey, why not 4? Wait, Baba. It could have thrown out 4, but it did not because it has used for the other 2H plus. Look at this. The electron that came from cytochrome C to cytochrome A3 went on to half molecule of oxygen. Getting it? It went on to half molecule of oxygen. So if half molecule of O2 gets 2 electron, won't it become minus minus? Right, na? And I want to negate it. So I gave it 2H plus. So what happens? The 2H plus gets cancelled. Correct? And half O2 means O becomes H2O. Hey, this is called as, you know what? Metabolic water. So who's the who's the proton acceptor? Oxygen. Who's the final electron acceptor? Oxygen. Oxygen has a double role. Huh? It is both an electron acceptor as well as a proton acceptor. Isn't it cool? Because everybody remembers that oxygen is the final electron acceptor. Nobody tells you that given right here is that oxygen is the final hydrogen acceptor also. Things cannot come out of your textbook. They come from the tech part of the textbook that you forget to read. Oxygen is the final hydrogen acceptor as well as the final electron acceptor. Are we clear? Beautiful. If you're clear with electron transport system, na, write in another comment. ETS rocks. Hashtag super simplified and tag me. Okay. Are we clear? Very, very easy. This chapter is like super duper simple. Clear. Very, very easy. Imagine now my check is FADH2. You know where it is made? Remember Kreb cycle? Succinate to fumarate step. See, succinate to fumarate. During this step, the energy liberated is not very high. It cannot be given to NADH plus H plus. So instead it is given to complex 2, FAD. The FAD takes the hydrogen. You know what the remaining story is. The hydrogen is split into H plus and E minus. The E minus goes to cytochrome B, iron sulfur C1, C, A, and A3. You know this, right? Hydrogens are pumped out. See here and here. Huh? Only two times. Only two times. NADH na pumps it three times. 1, 2, 3. FADH2 pumps it only two times. 1 and 2. FADH2 can only pump two times hydrogen outside via complex 3 and via complex 4. Getting it? Not via complex 2 because you know he is stunted. Okay? Not at all tall. That's why. So NADH is able to pump out hydrogen three times through three complexes, 1, 3, and 4. And complex 2 or succinate fumarate or FADH2 is able to push out hydrogen only twice, one with complex 3 and one because of complex 4. Are we clear? Hey, if you keep throwing such hydrogen outside, protons outside, don't you think there will be too much of proton outside the mitochondria in that intermembranous space? Yes. Don't you think the hydrogen want to come back along their gradient? Yes. Will they come back? No. Why will they not come back? Arey, the inner mitochondrial membrane does not allow them to come back now. Now, if they cannot come back, but they want to come back, let's allow them to come back now. But in exchange for their energy, these protons, as they go out, they have a potential energy. Let them come in. Let us keep a special complex 5 called as ATP synthase complex made up of two parts, F0 and F1. The F0 is like a tunnel. Getting it? It will allow that hydrogen, that proton, not hydrogen, proton, to come from outside to inside along the gradient. But with a special facility, getting it? It's not like they just crossed over. No. They did not require energy. They did not require ATP to cross in, but they required a special protein to do that. You had to make a special facility. Getting it? And that's why this hydrogen coming in is called as facilitated diffusion. Okay. So, facilitated diffusion. So let it come in. And as it comes in via F0, facilitated diffusion, you will allow that hydrogen ka energy, that potential energy to convert ADP plus phosphate into ATP. Now, as you can clearly see in this diagram, the rectified diagram in the 2025-26 reprint edition of your textbook, when 4H plus come in, 1 ATP molecule is produced. Super duper important. And as far as our textbook goes, because of NADH, 3 times hydrogen went out. So 3 ATP will be made, and because of FADH2, twice protons are pumped out, and hence 2 ATP will be made. You will remember it this way. Let us read this, let us mark all the important stuff, and then this video will end. The part 2 of the video will of course contain the remaining part of the chapter, which should be uploaded in a day or two. Electron transport system and oxidative phosphorylation. The following steps in the respiratory process are to release and utilize the energy stored in the NADH and FADH2. So why do you want the ETS and the oxidative phosphorylation? To release the energy stored in the NADH plus H plus and FADH2. So how do you accomplish this? Like, how do you achieve this? This is accomplished when they are oxidized through the electron transport system and the electrons are passed on to oxygen, resulting in formation of water. Remember what we said. Oxygen is the final electron acceptor as well as the final hydrogen acceptor. The metabolic pathway through which electron passes from one carrier to another is called ETS. A important MCQ statement. The metabolic pathway through which electron pass from one carrier to another is called as electron transport system. Where is it located? Inner mitochondrial membrane. The electrons from NADH produced in the mitochondrial matrix during citric acid cycle are oxidized by what complex? Complex 1, NADH dehydrogenase. You already know. The electrons are transferred to ubiquinone. Where is ubiquinone? Located in the inner membrane. Ubiquinone, if you recall, also gets the electrons from complex 2. So ubiquinone also receives reducing equivalents via FADH2. FADH2 is via complex 2. That is generated during oxidation of when is FADH2 generated? Generated during oxidation of succinate in the citric acid cycle. Succinate to fumarate. The reduced ubiquinone, MCQ, reduced ubiquinone is called as ubiquinol, is then oxidized. How do you oxidize something? Snatch its electron. Right? So you oxidize it with transfer of electrons to cytochrome C. Why cytochrome B, C1 complex? Where is cytochrome C? Cytochrome C is a small MCQ protein. Cytochrome C is a small protein attached to the outer surface of the inner membrane, and it acts as a mobile carrier for transfer of electrons between complex 3 and complex 4. MCQ, MCQ, MCQ. Cytochrome C is a small protein. It is on the outer surface of the inner mitochondrial membrane. It is a mobile carrier for transfer of electron between complex 3 and complex 4. Complex 4 is also called as cytochrome C oxidase. So simple, because it contains A and A3, two copper containing center, and they snatch the electrons from cytochrome C. That's why they are called as cytochrome C oxidase. MCQ, they have two copper centers. How awesome is the MCQs here? When the electron pass from one carrier to another via complex 1 to 4 in the electron transport chain, they are coupled to ATP synthase complex 5 for production of ATP. How? That hydrogen, the number of ATP molecules synthesized depends on the nature of the electron donor. Means. Oxidation of one molecule of NADH gives rise to 3 ATP molecules. One FADH2 molecule will produce 2 ATP molecules. Okay. Now, this entire chapter, we spoke about aerobic respiration. Have you noticed right till now, oxygen is not even there. Oxygen is limited to the terminal stage of the process. It's at the last. But without that oxygen taking the electron, or without the oxygen taking the hydrogen, how will the process continue? Somebody has to remove electrons and protons. Now, that's why the presence of oxygen is vital. It drives the whole process by removing. How does oxygen drive the whole process? By removing hydrogen from the system. Oxygen is the final electron and the proton acceptor. Okay. And remember, in photosynthesis, in some other video where we have discussed, it's the sunlight that causes ATP synthesis. Here, it is the energy of oxidation reduction utilized for the same process. Hence, we call this process oxidative phosphorylation. How easy is this chapter? Tell me now. Isn't it so simple? Now, in photosynthesis, you might have studied about the chemiosmotic hypothesis. If you know it, comment below, I know chemiosmosis. But if you don't know, please drop a comment so I can make a separate video for that. The energy released during electron transport, the energy that is released during electron transport is utilized in synthesizing ATP with the help of ATP synthase. Two major parts to this, two major components, F1, F0. F1 is the headpiece of this peripheral membrane protein complex. This is the site for ATP synthesis. F0 is an integral membrane protein complex. MCQs. F1 is a peripheral membrane protein complex. F0 is an integral membrane protein complex. F0 forms a channel through which protons cross the inner membrane. Simple. And as the protons pass through the channel, it is coupled. It is joined to the catalytic site of F1 component. And that's how ATP is made. MCQ again, for each ATP produced, 4H plus have to pass through F0, through the intermembrane space to the matrix down the electrochemical proton gradient. Easy, 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 easy. If you have understood everything till now, in the comment below, please write, I got this. So I know that you have understood it. And if you have doubts, please comment below so I can explain the doubts to you. Simple. Now, the next few minutes are not really a part of NEET, but I just wanted to clear certain confusion. You might have heard certain professors talk about how one NADH, H plus basically, is equal to 2.5 ATPs, and how one FADH is equal to 1.5 ATPs. But then again, during all the calculation, they do not take these numbers. They take the numbers 3 ATP and 2 ATP. Now, why this confusion? Well, honestly, they are correct about 2.5 and 1.5. But as far as our NEET is concerned, this information is practically irrelevant. I'll tell you where this number comes from, and you can understand it much better. They have clearly mentioned 4H plus have to pass to give you 1 ATP, right? So if you look here at this particular diagram of ours, you will realize that complex 1 pushes out 4H plus. Complex 1 pushes out 4H plus clearly. So this 4H plus will be in 1 ATP. Complex 2 again pushes out 4H plus, which means this is another ATP. So this is the first ATP, this is the second ATP. And complex 3, look at this. It only pushes out 2H plus. This literally translates to half ATP. Alright, and that's how the professors come up with this idea that one NADH must be giving 2.5 ATP and one FADH must be giving 1.5 ATP. But as far as our NEET is concerned, do not follow those numbers. Keep 3 ATP and 2 ATP in mind. It's good to be knowledgeable. It's better to be a doctor. So we are not focusing on being knowledgeable or intelligent. We are focusing on becoming doctors. I believe this video has been informative and must be able to make your chapter a little bit easier. The second part of the video is coming very, very soon. I will link it in the description box below. It will also be in a card link to this particular video. So you can easily click it and go to the new one and finish up this chapter. If you like this video, don't forget to share this video with your friends, subscribe to my channel, and I will see you with more informative videos in the next one. Till then, take care, keep studying, keep rocking, future doctors. Bye.